Good morning everyone. How are students? So students, in last class we understood about spherical mirror and we discussed mirror formula and we solved some questions also. We discussed in last class about real object, virtual object, real image, virtual image. Sign convention also we discussed. Okay, and by using all these concepts we solved some questions also. So students, here just quick revision. We discussed that if concave mirror is given, then this side all image are real and this side image are virtual. This side is positive, so V value, V is image distance is positive. If V positive, virtual image, V negative, then real image because this side negative. For convex mirror, all this side image are real and all this side image are virtual. This direction positive, so V value positive means virtual, V value negative means real. Okay, and we discuss about real object, virtual object. Okay, mirror formula we discuss 1 by V plus 1 by U equal to 1 by F. In this formula all values we have to put with sign. Okay, magnification we discuss height of image by height of object, formula is minus V by U. And here we discuss important point that for erect image meaning is what? Erect image meaning is object and image both form same side. Object, suppose if object is above the principal axis, then image also form above the principal axis, that is what erect. Inverted image means object is above principal axis and image formed below the principal axis, otherwise reverse. If object is below the principal axis, then image form above the principal axis, that is the meaning of inverted image. Today we will understand this point, then how to find that this image is erect or inverted without drawing ray diagram, by only solving question by using the data. How to know that this image is real, this image is erect or inverted, this image is real or virtual. So for real and virtual already we discussed, V value negative means it is real, V value positive means it is virtual. Now we will understand that by using data how to know that image is erect or inverted. Okay, for this we have to find out magnification. To know about erect and inverted image we have to find out magnification. Okay, so suppose we calculated magnification. Okay, now understand this point. We calculated magnification, this M. If this M value we are getting positive, if M value we are getting, two possibility are there. This M can be positive and this M can be negative. Agree or not? So if M is positive, if M is positive, then image is erect. Image is erect. Understood? If M is positive, then image is erect. You can ask, sir, how you can say that this image, if M is positive, then image will be erect. You can check. If magnification, height of image and height of image by height of object. We already discussed sign convention that above the principal axis positive and below the principal axis negative. Okay, so if object and image both are same side, suppose both are above, then height of image positive, height of object positive, both are positive, then finally magnification also positive. So if magnification positive, then image is erect. Now if magnification negative, then image will be inverted. How to know? You can check. Suppose object above and image below. Above positive and below negative. Below the principal axis negative. Okay. So one is positive and another is negative. Means overall it will be negative. Agree or not? Overall it will be negative. Okay. So here we can write that if M is negative, then image is inverted. Clear? So we understood about erect image and inverted image. Now next point, sometime we have to find out that image size is how much more than the object size, or image size is how much less than the object size, right or not? So how to know, how to find out? Understand, that is, we defined it that if magnification value is greater than 1, but understand here magnification can be negative, okay, because if image is inverted then magnification can be negative, so don't think that it is negative. So suppose -2 is there, m value is -2, so m value -2 means, it does not means that m value is less than 1. Here we defined mod m. What we defined? Mod m, because m positive or negative, it does not decide size of image is more than or less than object. M positive or negative, it decide only what? That image is erect or inverted. M value, mod m will decide image size is more than object or less than object. What will decide? Mod m. Mod m means if -2, so that also plus, we will use here +2. Mod -2 means 2. Mod -3 means 3. Mod m is greater than 1, then image is enlarged. Enlarged means image size is more than object. Okay. Mod m is less than 1 means image size is less than object. Image is smaller than object. Is it clear? So already I explained you that here mod m we have to take not m, because m positive negative, it decide erect and inverted image. To find out image size is more than or less than object, we have to check mod m, and mod m meaning is that if -2 is written, -2 mod is written, so -2 mod is equal to 2 only. Okay, and mod 2 is 2, mod -3 is 3, means negative value becomes positive by using mod sign. Is it clear? Okay. Now next point. We can say enlarged, here we can say magnified. Okay. So we understood how to find erect and inverted image, and we discussed that image size is more than object or less than object. How to know? By using mod m. Mod m is greater than 1, then it is enlarged, less than 1, then it is smaller. Okay. Now students, next. Yesterday we solved one question. Today we will solve one more question. This question. What is given? An object of height 1.5 centimeter. What is given? Height of object. Height of object. So this is HO. HO means height of object. So how much it is given? 15 centimeter. 1.5 centimeter. 1.5 centimeter. Okay. Is situated. So this is HO. This is what? HO. HO is given at a distance of 15 centimeter from a concave mirror. So which mirror? Concave. How much distance? 15. So what is given? U value is given. Okay. So here U value also given. This is what? U. But already I explained you that here we have to take negative, not positive. Okay. Why? Because this concave mirror is like this and we should put object in front of the concave mirror. Okay, so suppose here is the object. This object is placed like this. Okay, how much height? This is object. How much height? 1.5 centimeter. Height of object is 1.5 centimeter. This is HO. And distance of object is how much? 15 centimeter. Okay, so this direction, you know, this direction is positive and this direction is negative. Everything we calculate from pole. So we have to move from pole to object in this direction, and this direction is negative. So here U value is negative. U value is minus 15 centimeter. Okay. The concave mirror form its real image. So concave mirror form real image. You know, real image means which side? This side, है ना? Already we discussed. And for this side, V value is negative. Agree or not? Okay. Of height 3 centimeter. This is also given that height of image is 3 centimeter. So height of image is 3 centimeter. Okay. The focal length of concave mirror we have to find out. So what we have to find out? Focal length. Focal length means F value. Focal length we have to find out. Okay. So again, which formula we have to use? So only one formula we know about this mirror, that is 1 by V plus 1 by U equal to 1 by F. Okay. Some students are sending their answer. Fourth option they are sending. Okay. But you know, here in this question, negative value also given, right? So you have to take care everything. Okay, students. So one formula we have, that is 1 by V plus 1 by U equal to 1 by F. Agree? Okay. So we need to calculate F, we need U and V. U is given, yes, U is given. Okay. What about V? V given or not? So height of image given, height of object given, U given, but V not given, right? Now how to find out V? First we have to calculate V. U given, then only we can calculate F. Okay. So how to find out V? So in the question, height of object, height of image, these two quantities are given. So is there any relation between height of object, height of image and V and U? Is there any relation? Yes. What is that? Magnification formula. Right or not? Magnification formula. So magnification formula also we know, that is minus V by U, and this is height of object by height of image. Agree or not? Okay. Now here one important point is given, that this image is real. Image is real. Okay. Real image means this side. Agree. Now this side image, V value negative. Okay. So you know V value here negative. Negative, negative, positive. What about U? U also negative. So overall we will get here negative. Magnification negative means inverted image. Magnification negative means inverted image. Agree or not? Again understand. V value negative. Why negative? Because real image is this side and this direction negative. So this V value is negative. Okay. So negative and negative become positive. U value is negative. So finally here negative will remain. So magnification will be negative. If magnification negative means inverted image. Inverted image means object, if object is above the principal axis, then image will be below the principal axis. Right or not? So image will be below the principal axis. Okay. So below the principal axis, you know, above positive and below negative. So here our conclusion is that image is inverted. image must be inverted. This is our conclusion. Image must be inverted. You know, I am not discussing here direct and simple question. I am discussing here tricky questions in which you have to use your mind. These are not direct that put the v value and u value and you will get f. Right or not? So image must be inverted. Inverted image means height of image negative, height of object positive. Okay. So height of object how much? 1.5. Height of image 3. Okay, put the values. So here actually I did mistake. Here I wrote reverse values. This is height of image by height of object. So height of image how much? 3, but here we have to use minus. Okay, because it is inverted image. And height of object 1.5. Object above the principal axis, then image will be below, and below negative, above positive. So how much it is? 1.532, so minus 2. So minus v by u minus 2, so v by u equal to 2. Right? Minus v by u minus 2, so minus minus cancel out. So v by u equal to 2. Understood? Okay. Now we have to find out focal length. U value is given. How much? Minus 15. So v equal to how much? Twice of u, and u value is minus 15. Okay. So v value we got minus 30 centimeter. Now we can put all these values in this formula. Okay, what we have to find out? F. So 1 by v, v value is minus 30. Now understand this point. You know, this formula, earlier also I said that here whatever values you know, put those values with sign. You will get unknown quantity finally with sign. By default you will get with sign. So we are getting this v value with sign minus 30, and we know it is negative because image this side. Right or not? So formula eventually will give final value this unknown quantity with sign. No need to use here sign. Okay. Now 1 by v value is minus 30. Okay. And u value is minus 15. So minus 15 equal to 1 by f. Okay, solve it. So we can take LCM here, 30 minus, we can take common. So 30 is LCM. Here 1 and this is 2. Right? Equal to 1 by f. Now this is 3 by 30, 10 and minus sign. So f equal to minus 10 centimeter. This is our final answer. So first option is correct. Many of you were getting fourth option. Finally, many students said this answer, first option. Okay. So we solved one question, but this question is how many concepts? How many concepts we use here and how much we learn by solving one question only. So this is for development of the students, right? By using each word, I explained you that what is the meaning of this word. This word is given, this word real image is given. So real also has meaning here. What? That if real image means image this side, if image this side means v value negative. If v value negative, u also negative, both become positive, minus will remain. Magnification negative means inverted image. If inverted image, then height of object 1.5. If we take an object above the principal axis, then 100% image must be below the principal axis because inverted image. So in case of height of image, here we do not have to use 3 centimeter. We have to use here sign also, that it must be minus 3 centimeter because it is inverted image. Clear? So these are the conclusion. These are the points that we extract from each word of these questions. Like that you have to solve question in exam. Okay students, so I hope you all understood it. Now you have to solve this question. Okay, which points are given here? A boy stand straight in front of a mirror at a distance of 30 centimeter away from it. He sees his erect image whose height is 1/5 of his real height. The mirror he is using is, okay, so which mirror he is using here, we have to find out. So plane mirror you all use every day, right? You can do this experiment also. You are standing in front of plane mirror. So have you see your height is less or more? No. We see our actual height. Agree or not? So image height and object height both are equal if it is formed by plane mirror. So plane mirror form same size image, right? So plane mirror form same size image. But here what is given, that height is 1/5 of his real height. Okay. So this is not possible by plane mirror. Now 100% our answer can be convex or concave. Among these two options we have to choose any one. Okay. So it is concave or convex. How to find out? To this point also given that image is erect image, right or not? Image is erect image. And one more point given, what? That height of image is less than object. Height of image is less than object. So it is possible by which mirror? Concave or convex? Convex mirror is the answer. Okay, how? Understand. Without solving anything, how to find out it? Understand. So first point, it is erect image, है ना? Erect image means object and image both are in same side. Okay. Now erect image means magnification positive, right or not? Magnification positive. To understand this point here, they given that erect image. If erect image, so what is our conclusion? That magnification greater than 0, positive. Okay, magnification positive. Don't use here greater than 0. Just only we will use that magnification is positive. Now if magnification positive, and we know magnification formula minus v by u, and it is positive. Object is in front of the mirror, to object distance negative. Okay, so u value is negative, right? Here negative, so v value must be positive. Then negative become positive. If v, and finally we will get v value positive. Then u is negative, it is given. So our conclusion is that v must be positive. V must be positive. Now if, and one more point is given, what? That size of image is less. Size of image is less, magnification it is diminished image, mod m less than 1. Okay. So this is also given that mod m less than 1, because size of image is less, diminished image. Direct theoretical point is also there, but I am not using here any theoretical point. By giving values, how to find out? That question we can solve only just by the theoretical point. But I am teaching you that how to find out everything by using these formulas. Numerically, mathematically, how to find out. Okay. So understand it. So mod m is less than 1. Okay, it is diminished image. Okay. Now mod m is less than 1, so we know m is minus v by u. Okay, so minus v by u is m, so mod m less than 1, so we can say this negative also become positive because it is mod. So mod v is less than mod u, means image distance, this is what? Image distance is less, object distance is more. Agree or not? Okay. Now use mirror formula. Okay. By using mirror formula, 1 by v plus 1 by u equal to 1 by f. Right? This thing we know. Mod v is less than mod u. Okay. So for which case it is possible, understand. So do one thing here. Transfer this u this side. So it become 1 by v equal to 1 by f minus 1 by u, take LCM. So u minus f by f into u is 1 by v. Right? So v equal to how much? F u by u minus f. Now divide this u this side. So v by u equal to f by u minus f. Now you can understand here. Focal length is negative for which mirror? Concave. Okay. So negative, negative, positive. U value negative. Right? So we are subtracting something. So this value or all this value. Again listen. U value is negative in all. Okay. It is negative. F value focal length is positive for convex, negative for concave. Okay. So take for convex. For convex F value positive. So here it will be negative only and u value is negative. So both are negative, negative, both will be added. So this value is greater than f. Right or not? So u minus f is greater than f for convex. So if it is greater, then overall v by u is less. Right or not? So v by u, mod v by u is less than 1, means diminished image. Agree or not? So means diminished image. Okay. And for concave, f value negative, 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 positive. So 1 is positive and 1 is negative. So overall this value is less than this value. So if it is less, then overall we will get greater than 1. So v by u is greater than 1, means in large image. Why? Because u minus f is less than f for concave mirror. Why it is? Because u minus f is less than f for concave mirror. Clear? So here ultimately what we learned a point by this numerical method. Finally now we can write this theoretical point and we can remember it, but I proved it and you understood all these points. Okay. So finally what you have to remember, that if image is erect and magnified, then it is formed by concave mirror. Okay, this is golden point, key point that you have to remember. Second, if image is erect and diminished, then it is formed by convex mirror, right? So now we understood the theoretical point. First we find out everything numerically. By numerically we understood, then we make a point here, theoretical point. Now we will remember always that if image is erect, then it is, and image is erect and magnified, enlarged, then it is formed by concave. Image is erect and it is diminished, then it is formed by convex. Is it clear? Okay, so students, here this question answer now we can give without solving anything, convex mirror. Is it clear? So we understood this question today, and in this question we understood so many concepts, how to apply each concept, that also we discuss, and by using each word I explained why it is given, what is the sense of this word here in this question. So these points I explained today, and we discuss about magnification, positive, negative, that also we discuss. Okay, magnification positive, then erect, negative, then inverted, mod m greater than 1, then image enlarged, less than 1, then it is diminished image. Okay, and mod meaning I explained, -2 is 2, means negative sign become positive after using mod. Okay friends, so now I can assure you that you can solve any questions related to this mirror equation, convex or concave spherical mirror. Okay friends, so I hope you all enjoyed this session and you understood all these topics. Okay.
Good morning everyone. So yesterday we have seen how to balance redox reactions. One is when all neutral species are there, how to balance. When ions are there in an equation, how to balance. These two things we have seen. When ions are there, one additional step you have to take care, that is charge balance. Once the cross multiplication step is over, next is charge balance step. If H plus ions you are using to balance, then add it to the lower side, lower charge side. If OH minus you are using, add it to the higher charge side to balance the charges. Let's see a few more examples and then we will go to the next topic. In the reaction 8 aluminium 8Al 3Fe3O4 gives Al2O3 and Fe. The number of electrons transferred from the reductant to oxidant is, how many electrons are transferred? Now see, what is the oxidation state? If you take aluminium, I am writing the equation again, Al plus 3Fe3O4 4Al2O3 9Fe. Okay. First elementary states, oxidation states, let's write. Elementary states, oxidation states are 0. Iron also elementary state 0. Now oxygen -2. When oxygen is -2, what will be the oxidation state of aluminium? You will get +3. Then only see, aluminium anyway always +3 only in compounds. Oxygen -2. And here oxygen -2 if you take, what should be the iron oxidation state in this? Let's figure out Fe3O4. If oxygen is -2, iron is X. 3 irons are there. So 3 into X. 4 oxygens are there. 4 into -2 is -8 is equal to overall charge of species 0. So X is equal to 8/3 you are getting. So iron oxidation state is 8/3. Now, see, you take aluminium or iron, it does not matter. You will get the same answer because whatever electrons involved in oxidation, whatever released through oxidation, those electrons only will involve in the reduction side. So whether you take oxidizing species or reducing species, you will get the electrons involved will be same only. They only transfer from one place to other place. So here, I'll take aluminium to calculate the electrons. For one aluminium, how many electrons are involved? What is the change in oxidation state? That is the number of electrons involved. Electrons are only responsible for the change in oxidation state. What is the change? 0 to 3. 0 to 3 means 3 electrons involved for 1 aluminium. For 1 aluminium, 3 electrons involved means how many aluminiums are there in total? Actually 8. Here also see 4 into 2, 8. So 8 aluminiums are involved overall. For 1 aluminium atom, 3 electrons are involved. 8 aluminium atoms, how much total then? 8 into 3. So 24 electrons are involved in total. The same 24 will be, if you take iron also, total number of irons if you take, there also you will get the 24 only. If you want, you can try later on. So answer is 24. Balance the equation. We have to balance this equation. Now see, first write, let's oxidation state, oxygen -2, manganese +7. I am writing for those elements which are undergoing change in oxidation state. Rest elements I am not putting, only those elements which are undergoing change in oxidation state, only I am writing here. Please understand that. So oxygen +4, you know how to calculate oxidation state of sulfur. Take oxygen -2 and just like previous case, you take the overall charge and equate it to the charge -2 and find the sulfur oxidation state. So like this, which are undergoing change only I am writing. Manganese is undergoing change, sulfur is undergoing change. So 1 manganese, 1 sulfur, 1 sulfur, already balanced. Now what is the change? See, 7 to 2, change is 5. 4 to 6, change is here 2. So ratio is 5:2. So you have to multiply number 5 with the sulfur items, number 2 with the iron items you have to multiply. So let's multiply that. I am writing this here directly only, or I will write one more time. So let's put the numbers. So manganese items you have to multiply with 2. 5 you have to multiply with the sulfur items. 5, 5. Okay. Now next step is, since it is an ionic equation, charge balance. Now what is the total charge on the left side? Don't take H plus into consideration. Using this only we have to achieve the balance. So here 2 into -1, -2, 5 into -2, -10. So total -12. Right side 2 into +2, +4, 5 into -2, -10. So it is -6. So charge is not balanced, we have to balance it. So what is the difference? -12, -6, difference is 6. Difference is 6. So 6 H plus we have to add. Already H plus is mentioned left side only, we have to add left side only because lower charge is -12 only. Add 6 H plus. So H plus will be there for 6. That's it. So 2 MnO4 minus 5 sulfates, 5 sulfides plus 6 H plus, 2 Mn plus 2, 5 SO4 minus 2, and water you can balance because 6 H2O, 6 H plus ions are there, means to balance we have to put 3 H2O there. Automatically everything get balanced. Is it understood, everyone? Okay, next we will see next topic, quantum numbers. See, there are 4 quantum numbers. Knowledge of these quantum numbers helps you to write the electronic configuration of various elements. Now, what are these quantum numbers? There are 4 quantum numbers. One is principal quantum number. Denoted by n. Second one, azimuthal quantum number. Denoted by L. Third one, magnetic quantum number. Denoted by ML. and fourth one spin quantum number. Denoted by ms. See, how to understand these quantum numbers, what they are actually, what is the meaning of them with respect to electrons. See, look here, these four are like this. Suppose if you go to some courier center, suppose if you want to send one courier to your hometown or somewhere, you go to courier office, take a parcel and put the items, everything in that parcel, and you have to write the address. Now, in place of address, if you write the Aadhaar number of the address of the targeted person, Aadhaar number, and you give it to the shop person, courier person. Now, you can understand how he will look at you. So, Aadhaar number, actually in government sites, if you put the Aadhaar number, you will get all the details, person name, you will get everything, when he or she born, and what is the address presently he is living, everything, all details you will get when you enter the Aadhaar number in the website. But if you give this Aadhaar number to the person, courier person, will he accept it? Will he search and note down all the details? No. He will just write it off and he will scold you also if possible. Then he will ask you to write the actual address. Then what you will write? You will write the city where the person is living, to which courier has to be sent. And in that city there may be many streets will be there. You have to write the street name also. In that particular city there may be many streets, street name, and in that street there may be many houses. You have to mention the house also. So house number. And in that particular house there will be many people. You have to mention the name of the person in that house. So like this you will mention on the courier parcel. Then he will accept, he will take and he will send. So for a particular person, if you are addressing, then these details are required. Similarly, in an atom also, there will be many electrons will be there. All are electrons only, like how all are human beings only in this land. So similarly in atom also there will be various electrons will be there. All are called electrons only. Suppose if you want to distinguish one electron from other electron, if you want to call one electron, then something will be there, something you have to use. If you just call electron, then all electrons will look at you. Then how to distinguish one electron from other? So some address should be there for each electron. Now that address is principal quantum number, that is n, azimuthal quantum number L, and magnetic quantum number ml, and spin quantum number. Principal quantum number is like city name. Imagine there is in atom, cities are there, many cities are there. Okay, if you are calling one electron first, you are addressing one electron. Okay, you are discussing about a particular electron. Then which electron? You have to mention first city, in which city that electron is living. We don't call it city here, we call it principal quantum number, n. Now in that city or in that shell, there will be many streets will be there. Electron is living in which street? Which street electron you are talking about? That you have to mention. That is azimuthal quantum number. It is similar to the principal is similar to city, analogous to city. Azimuthal is analogous to what? Street. Now in that street, like how in normal streets many houses will be there, similarly in an atom, in that particular street, there will be many houses. So which house electron you are talking about? You have to mention the house number also. So that is like magnetic quantum number. Now, magnetic quantum number is like a house number. We call here in atom the house, we don't call house, we call orbital actually. So it is the orbital number or magnetic quantum number. Now, in that particular house, in an atom, persons, maximum 2 persons will be there, male and female. Actually, normally in a family there will be many multiple persons possible, but in an atom, in a particular house, maximum 2 persons will be there. One male, other cannot be male, other has to be female, male and female. Or only one person can be there also, male or female. or no person also can be there. House can be empty also, but not more than 2 persons. If there are 2 persons, one has to be male, one has to be female, like that. Imagine, I mean, visualize like that. So, in other words, here, one electron will be in clockwise motion, other electron will be, spin will be in the anti-clockwise. That is what male and female analogy I am talking about. So, if you are addressing a particular electron, spin, there are 2 electrons will be there in an orbital or house. So, which electron you are addressing? Okay, male electron or female electron? Spin, clockwise spin or anti-clockwise spin. So, that also if you mention, then you are calling a proper electron, particular electron you are calling in the atom. You have mentioned city, in that city which street, which house number, in that house which electron you are calling. When these 4 are mentioned, then only electron is defined. Electron name is defined. You can say, you can address the electron in an atom. That is what quantum numbers meaning actually. The quantum numbers, what they actually mean, what they actually represent. These 4 quantum numbers together, address the electron in an atom. If any one quantum number is changing here, you are talking about a different electron. You are not talking about the same electron. So is it understood everyone what actually they mean in terms of analogy? Okay. Now, these 4 quantum numbers, first we'll take the principal quantum number. Now, here we don't call here city, in place of that we call shell here in an atom. So, principal quantum number is actually a shell number. So, it represents and here we represent shells with a numbers 1, 2, 3, 4 like that. So, the cities in the atoms are represented as a numbers 1, 2, 3, 4 like that. So, n values starts with 1, 2, 3, 4 like that. Now, in that particular city or shell, we call it here in atom. So, there will be many streets. That is azimuthal quantum number. Let's take that azimuthal. Now, this azimuthal quantum number, it represents subshell. We call it here street. We don't call it street here. We call it the word subshell. Shell, inside that there will be many subshells will be there, like how in a city many streets, here also many subshell. So, that is a subshell. So, azimuthal quantum number is actually a subshell number it represents. And what are the values we give? We starts with a 0, L is equal to 0. Actually, in a city, streets have names like Rajiv Gandhi Colony, Ambedkar Colony, like that names will be there. But in an atom, the streets have numbers. With numbers, we will define the street. And the number starts from 0, 1, 2, up to n minus 1. N is the principal quantum number. Suppose principal quantum number is 3. Then 3 minus 1 is what? 2. So, there will be streets like 0, 1, 2. That's it. Only 3 streets will be there in that third city, not more than that. Like that, L values depends on the N value. Next, one more thing here. See, these numbers, principal quantum number, N is equal to 1. N is equal to 1 is called K shell. Alphabetically, it is called K shell. N is equal to 2 is called L, M, N, like that. These are the alphabetical notations of the shells. K shell means principal quantum number 1. N means principal quantum number 4, like that. Similarly, here L is equal to 0. L is equal to 0 is called S subshell. L is equal to 1, P. L is equal to 2 D, L is equal to 3 F. Like this, and these are the numericals, numbers for a subshell, and they are also given as alphabets like this S, P, D, F. These are the subshells. And so far only up to F subshell only we have seen whatever elements, 118 elements we have in periodic table. No element has beyond F. Suppose in future new elements are discovered which demand higher subshells, then we will take the next subshell G. We will define that as G, E, F, G, H, like that it will go. So presently only up to F only, SPDF subshells only available for the whatever elements we have discovered so far. Okay. Next, third thing, magnetic quantum number. So city, street over, next come to the house number, which is magnetic quantum number, ML. Now, this magnetic quantum numbers, it represents orbital. City we call in this case shell, street we call subshell, house we call in atom corresponding word is orbital. Orbital is the actual, imagine like a house of electrons. Inside that orbitals, electrons will be living, like that you visualize. Now it represents orbital, and these values here, orbitals also have, like how houses have numbers, similarly here orbitals also have numbers, like cities, shell numbers, subshell numbers. Here orbitals also have numbers, and those numbers will vary from minus L up to plus L, minus L to plus L. What is the meaning of it? Suppose, example, if L is equal to 2, subshell number is, or street number is 2, then in that street or subshell, how many houses will be there? That is orbitals will be there. ML value starts from minus L. Minus L is what? Minus 2. And next number is what? In the scale, in the number scale, what is the next number? After minus 2, minus 1, 0, 1, and 2. That's it. You have to start at plus L value, you have to stop there. Up to 2, you have to take. So these many houses or orbitals will be present in the subshell, not more than that. How many? 1, 2, 3, 4, 5. 5 orbitals will be there. Next, fourth one is spin quantum number. So spin quantum number talks about spin of electrons. If electron has a clockwise spin, then first it represents spin. Now clockwise spin, then ms value will be taken as plus half. Anti-clockwise spin, ms value will be taken as minus half. That's it. Clockwise spin plus half, anti-clockwise minus half spin. These are the 4 quantum numbers. Now let's take here N, which represents shell, L, which represents subshell, ML, orbital. Okay. Next is spin. Spin anyway, 2 spins are only there in every orbital. Now here, suppose if you take N value 1, it cannot be 0. N values we have seen, it begins with 1 only. If N value is 1, L values will be what? L value starts from 0, end with N minus 1. N minus 1 is 1 minus 1 only, that is 0. It starts with 0, ends with 0 also. So only one street will be there, subshell will be there. And ML values what? ML values starts from, L value starts from 0 and up to N minus 1. ML value starts from minus L, ends with plus L. So minus 0 to plus 0. That means only 0. So in the city number 1, there will be only one street, and in that street only one house will be there. What a lonely the house is. Okay. And next, suppose if n is 2, L value starts from 0, n minus 1 is what? 2 minus 1, 1. So it starts from 0, ends with 1. And ML values here, 0 means in that street only 0, one house only will be there, minus 0 to plus 0. When you come to this subshell, minus L to plus L means what? Minus 1, 0, 1. 3 orbitals or houses will be there. Next 3, 0, 1, 2. Because n minus 1 is 2, so start with 0, end with 2. For 0, only 0 will be there. For 1, minus 1, 0, 1. For 2, minus 2, minus 1, 0, 1, 2. These are the orbitals present in that subshell. So like this, we keep on going. Now, if you look at this, in the first one, only one subshell is there. In the second one, 2 subshells are there. In the third one, 3 subshells are there. So can I write number of subshells in a shell is equal to n value. Number of subshells present in a shell is equal to just value of n only. Here value of n is 1, that's why only one subshell. 2, 2 subshells. 3, 3 subshells. Next. Now come here. Now in a subshell, there are various orbitals are there. Suppose in first subshell, 3 orbitals are there. Second subshell, 1, 2, 3, 4, 5 orbitals are there. So how to connect them? See, number of orbitals in a subshell is equal to 2L plus 1. 2L plus 1. Because minus L to plus L means for one side of 0, L values will be there. Other side also L values will be there. 2L total. Including 0, total 2L plus 1 values will be there. See, L is equal to 2. Let's substitute L is equal to 2 in that 2L plus 1. You should get 5. Are you getting or not? 2 into 2 plus 1, 5. Yes, 5 are there. Put L is equal to 1. L is equal to 1 if you put 2 into 1 plus 1, 3. Yes, 3 orbitals are there. So like this, number of orbitals in a subshell is equal to just 2L plus 1. Third, number of orbitals in a shell is equal to n square. See, first we have see the relationship between these two. Second point, these two. Now this and this we are relating. Because in solving problems, sometimes they will ask like that. So first one and third column. See, in first shell, how many orbitals are there? 1. In the second shell, how many orbitals are there? Count total. 1, 2, 3, 4. In second shell, 4 orbitals are there, which looks like a square of 2. Square of 2 is what? 4. Now come to the third shell. Third shell, how many orbitals are there? Count. 1, 2, 3, 4, 5, 6, 7, 8, 9, which looks like 3 square. So just n square, shell number square, will give us how many total orbitals present in that shell. And that's it. Tomorrow we will see problems on this. How to find, see, whatever the points we discussed now, 1, 2, 3. So on those things, how problems are asked in exams, especially NEET exam, what previously questions are asked on this. Only with this information, many questions you can solve of the previous years. Okay, tomorrow we will see some of the questions. Thank you.
Good morning, students. Good morning, Ra. Today our botany class, pteridophytes. Actually, it is the middle group when compared to the lower plants and higher plants. After algae, bryo, next is the pteridophytes. After pteridophytes, next occupants are gymno and angiosperms. For example, this entire square is a representative for a plant kingdom. Representative of plant kingdom. In that primitive group is algae. Second occupants in this plant kingdom, bryophytes. Third occupants, pteridophytes. Fourth and fifth respectively, gymno and angiosperms. Gymno and angiosperms. It is the order in course of evolution. First evolved organisms within the plant kingdom, algae. Recently evolved one, and dominant plants in the planet at present day, angiosperms. Dominant plants in plant kingdom at present day are angio. Highly evolved angio. Highly primitive algae. Middle one, pteridophytes. I would like to explain one more important concept, but before going to that one, you should learn what are the two phases in life cycle of the plant body, or two generations in life cycle of plants. Already learned in previous classes. Once recollect or remember, name the two phases in life cycle of the plant body. Now give answer. Try to recollect. Good. Amaravati, Harika. Good. Gametophytes, sporophytes. Nice. Balu, good. Gayatri, good. Gametophytes and sporophytes. Can you differentiate the gametophyte and sporophytes? One of the phase is the gametophytic generation, another one is the sporophytic generation. Okay, good, very good. How can you differentiate? How can you differentiate gametophytic body from sporophytic generations? Very good, excellent. Gametophyte is in haploid condition N. Sporophyte is in diploid condition 2N condition. It is a major variable variation or differentiation between gametophyte and sporophyte. Definition for the gametophyte is the gamete producing phase or generation, gametophyte. Spore producing generation is the sporophyte. Variation is it is a haploid, it is a diploid. Yes, one more question regarding this life cycle. In these two phases, which one is the independent, which one is the dependent? The phase which can perform photosynthesis, that is independent. The phase which is depending on another phases for the food material, that is the dependence. Gametophyte is dependent or sporophyte is dependent? I'll ask one more kind. Gametophyte is independent or sporophyte is independent? So independence and dependence nature of gametophytes and sporophytes variable from plant to plant. Variable from plant to plant. It is one of the major variation between all the plant groups in this plant kingdom. In algae, for example, it is the life cycle in algae. Algae life cycle. Gametophyte is independent. What is the meaning for independence? It is the green color, photosynthetic. It is able to prepare food material one. For independent gametophyte, the representative color is the green. Due to the chlorophyll nature or photosynthetic nature, it is independent. It is a dependent. In algae, gametophyte is independent or photosynthetic. To explain this concept, I can use one representation here. It is a haploid gametophyte. Again, haploid gametophytes. These are diploid sporophyte. 2N, 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 2N. Identify the colors. Where the green coloration is there, that phase is independent or photosynthetic. Independent or photosynthetic. Based on this kind of graphical presentation, you can solve several questions here. Several questions. What is the independent phase in algae? In algae, green color is given for N. So gametophyte is independent. Gametophyte is independent. What about the bryophyta? In bryophyte also, gametophyte is independent, but this black color portion of the sporophyte is dependent. Sporophyte is unable to prepare food material. That's why sporophyte is dependent on gametophyte. In bryophytes. What about pteridophytes? Yes, in pteridophytes, both the phases are independent, gametophyte and sporophyte. It is the unique feature of pteridophyta plants. Unique feature, peculiar character of pteridophytes. In entire plant kingdom, this kind of nature is found in pteridophytes only. Both the phase generations, gametophytic generation as well as sporophytic generation, both are independent, both are chlorophyllous, both are photosynthetic. Both are photosynthetic. That's why both the phases are independent. But the story is reversed from gymnosperm onwards. Observe here, gametophyte is in black color, unable to prepare food material, so on. That's why it is dependent, but sporophyte is independent. Even in angiosperms also, gametophyte is dependent, sporophyte is independent. Okay, from based on this compared to chart, we can make some questions. Ready to give answers for the questions. Question number first, name the plants. I will give options first of all. First option algae, second bryo, third pterido, fourth and fifth gymno and angio respectively. 5 options are there for this question. In what plant groups sporophytes are independent? Question number 1. Sporophytes are independent in. Independent sporophytes are found in. Independent sporophytes are found in. Good. 3, 4, 5. Pteridophyte, gymnosperms, angiosperms, sporophytes are independent. Good. Nice. 3, 4, 5. Second question. Identify the plant groups in which gametophyte is independent. Independent gametophytes are found in. Yes, right answer. Algae, bryo, pterido, gametophytes are independent. Yes, 1, 2, 3 is right answer. Next, one more question. Question ready? Dependent gametophytes are found in or non-photosynthetic gametophytes. Now, answer for this question. 5 options are ready. Choose options. Yes, good. 4 and 5. 4 gymnosperms, angiosperms, 5. Next, one more question. A slight change. Dependent sporophytes are found in which plant groups of the plant kingdom shows the possess dependent sporophytes. Dependent sporophytes are found in 5 options. Good. 1 and 2. Yes, nice. 1 and 2. In both algae and bryophyta, gametophyte independence, sporophytes are dependents. Okay, good. Nice. Next, one more question. Both sporophytes and gametophytes are independents. Both the phases are independent in. Choose the right answer. Yes, good. In pteridophyte only, total entire column is in green coloration. Both the phases, sporophytes and gametophytes are independents. Good, nice. Yes, yes, yes. Next, one more question. This concept is slightly different. Observe the diagram carefully. Be ready to give answers. What is the difference between this pink color area and brown color areas? Here, who is the, what is the name of this pink colored portion and brown portion? Pink one is gametophyte. Brown one is sporophyte. In course of evolution from algae to angio, gametophyte is in reduction or progressive? Progressive evolution is exhibited by gametophyte or sporophyte? progressive evolution exhibited by gametophytes or sporophytes in terms of the evolution from algae to angio, which generation of the plants can exhibit the progressive evolution. Progressive evolution. Yes, sporophyte. Observe this diagram clearly. In algae, very little portion is there in the sporophyte. Bryophyta, something enlarged. Pteridophyte, enlarged. Gymno, very well enlarged. Maximum enlargement is there or progress is there in sporophyte. So, in course of evolution, sporophytic generation can exhibit to progressive evolution or progressive development. Progressive development. What about the gametophyte? Reduction in their progress or retrogressive evolution. This kind of evolution is called retrogressive. Reduction in progress, nothing but retrogressive evolution. Reduction in progress. In course of evolution, gametophyte can exhibit reduction in their progress. Sporophyte can exhibit progressive evolution or progressive development. Now, based on these concepts, I can make some few questions or simple questions and elementary questions. Options for 5. Algae. Second option Bryophyta, third Pteridophyta, fourth Gymno, fifth Angio. Now the question is, options ready before the question. Question is, highly developed sporophyte is present in or is found in. Question ready? Highly developed sporophyte is found in or well developed. Extensive sporophyte is found in. First place goes to angio. Next place gymno. In both gymno and angio, highly evolved sporophyte is there. Within these two groups, angiosperm is a more elaborative sporophyte. It has more elaborative sporophyte than that of gymno. Okay, 5. Right answer. Good, nice. Next question. Highly developed gametophyte is found in. Highly developed gametophyte is found in. Good. Algae, very well elaborated. When compared to the size of the gametophyte, sporophyte is highly reduced or limited to only one cell, that is the zygote. In entire life cycle of the algae, zygote is the representative of sporophyte. Single cellular sporophyte is found in algae. Single cellular sporophyte is found in algae, except that zygote. Remaining entire life cycle belongs to gametophyte generation. Bryophyte also gametophyte is developed. Okay, but according to this question, the best option is the first option only. Next place goes to bryophytes. Okay, good. Next, one more question. Slide change. Highly reduced gametophytes are found in. Highly reduced gametophyte is present in. Reduced gametophyte. Good. Fifth one is the right answer. Observe here. Brown color portion occupies the maximum area, but this pinkish area is very smaller. It means it clearly explains gametophyte is highly reduced. Highly reduced gametophyte is found in angio. Good. Next, one more question. You can guess that question also. Highly reduced sporophyte is found in. Highly reduced sporophyte is found in. 1. First option, algae. Sporophyte is limited or restricted to the only one cell, that is the zygote. Remaining entire life cycle is in gametophytic condition. Good, good, nice. This is the compared account of all the plant groups in plant kingdom. Better to note this kind of presentation. Now, return to topic, pteridophytes. Now, what type of plants are included under pteridophytes? Mainly 3 groups are there in pteridophytes. In algae, 3 groups: brown algae, red algae, green algae. Likewise, in pteridophytes also 3 groups. Club mosses, these plants are called club mosses. Horse tails, nickname of this horse tail, commonly found in sandy areas. Generally, sandy locations are found the seashores, just at the edges of the rivers, and canals also at the bottom. Sandy location is there. In summer season, completely water drains away, only sand remains. That area is a more prominent area for growth of horse tails. Third one is the fern, ornamental plants. These are ornamental potted indoor plants. All these are pteridophytes. There is no flowers in pteridophytes. Non-flowering plants like algae, bryophyta. Pteridophyta is also non-flowering plants, cryptogams. But these are the first true land plants. Have to highlight this point. First, land plants are bryophytes. But first true land plants are pteridophytes. A slight variation is there. First true land plants, pteridophytes. First land plants, bryophytes. It is a slight variation between bryo and pterido. Economic importance of pteridophytes. Majority of the pteridophyta plants are used as one of the ingredients preparation of many Ayurvedic and homeo medicines. Ayurvedic and homeo medicines. And soil binders to prevent soil erosion. It is the spraying of spores of the pteridophytes on normal plain areas where the annual rainfall is more, floods are more. In that area, to prevent soil erosion, have to culture or propagate pteridophyta plants, because the root system of pteridophyta plants can hold or keep the soil, and such are called soil binders. Such roots are fixes in soil very strongly. Soil erosion is avoidable by using such kind of plants. It is the economical importance of pteridophytes. One is the medicinal purpose. Almost all the members of pteridophytes are medicinally beneficial. So that's why these leaf extraction of pteridophytes are used as one of the ingredient in preparation of Ayurvedic medicines. Soil binders. They are also frequently grown as the ornamental plants, indoor plants, potted plants. Evolutionary, they are first terrestrial land plants. And one important concept here. First vascular plants in the entire plant kingdom are first tracheophytes or pteridophytes. Due to presence of vascular tissues, these plants are called tracheophytes. Phyte, group of plants, tracheo, vascular tissues. These are first in entire plant kingdom. And these are embryophytes because embryo is formed in life cycle. Female sex organ is archegonia, that's why archegoniates. Vascular tissues are present, that's why these are tracheophytes. No flowers, that's why these are cryptogams, non-flowering plants, also called as cryptogams. Non-flowering plants. Compare the bryophytes to pteridophytes. Bryophytes also embryophyte, archegoniates, but atracheophytes, no vascular tissues. It is one of the major variations between bryophyte to pteridophyte. Bryophytes non-vascular, but these pteridophytes are vascular. Out of these 4 options, 3 options are similar in both bryo and pterido. Both bryo and pterido, embryophytes, archegoniates and cryptogams. But bryophytes exclusively atracheophytes, but pteridophytes are tracheophytes. Okay. Next point. What is the common habitat of the pteridophytes? Like bryo, here also very cool area, damp soil, shady locality, essential for the growth of pteridophytes. But sandy soil condition is a very well favorable area for growth of pteridophytes. Habitat completed. Habitat of pteridophytes. Next point, you may recall that in bryophyte, dominant phase in the life cycle is the gametophyte. Let's recently learn most dominant phase in the life cycle of the both bryophytes, gametophytic phase. The plant body in bryophyta is gametophyte, but in pteridophytes reverse. In pteridophytes, Dominant phase is the sporophyte or plant body is the sporophyte. Pteridophytic plant body is sporophyte, but bryophyte or plant body is gametophyte. It is also another comparison or differential character between bryophyta to pteridophyte. Pteridophyta organs. What are organs? Root, stem, leaves. Root, stem, leaves are the organs with vascularized nature. Means possess vascular tissues. All these organs are vascularized in pteridophytes. The root system of the pteridophyta plant is adventitious. It is the shape of the plant body in pteridophytes. Plant body differentiable into three parts, or sporophyte differentiable into three parts. One is the root, second entire portion is the stem, third one is the leaf. Well differentiated roots, stems, leaves are found in sporophytes. One of the examples for pteridophytes, polypodium. It is the sporophytic structure in pteridophytes. When compared to angiosperms, one organ is missing here. That one, no flowers. Due to the absence of flowers, seeds and fruits also absent. Except these three organs, remaining three organs are present. Root, stem, leaves are found in sporophyte of pteridophytes. Remaining three organs, flowers, seeds, fruits absent. Yes, root system is adventitious. Stem is a normal, very smaller, but leaves in pteridophytes are smaller. Small leaves found in Selaginella plant. Selaginella is one of the examples for pteridophyte. But larger leaves are found in ferns. So two types of leaves are found in pteridophytes. Smaller leaves, microphyls are found in Selaginella. Macrophyls means larger leaves are found in ferns. Phyll, scientific name of leaf, macro, larger. Phyll, leaf, micro, smaller leaves. Name the pteridophyta with small leaves, Selaginella. A pteridophyta members with large sized leaves, big sized leaves, ferns. Fern is one of the dominant group in pteridophytes. Around 80 to 70% of the pteridophytes belongs to this category, ferns category. Fern. Next, sporophyte of the pteridophyte bears sporangia. Such sporangia producing leaves are called sporophylls. If there is no flowers, to compensate absence of flowers, leaves involved in production of sporangia. Such sporangia producing leaves are called sporophylls. Phyll is the meaning of leaf. Spore producing or sporangia bearing leaves are sporophylls. First sporophylous plants in this plant kingdom, pteridophytes. These are helpful for sexual reproduction. But sexual reproduction, flowers are required. Without flowers, the sexual reproduction is possible by producing these spores. So sporophylls are an alternative structure for flowers. Absence of flowers in pteridophytes compensated by producing sporophylls. These sporophylls are either independently arranged or aggregate on one axis to form a cone or strobile-like structures. Each and every one of the structure is the sporophylls to form finally one compact structure. The name of this compact structure is cone, or one more name of the cone is a strobile. Another name of the cone is a strobile. What is cone or what is strobile? Now give the answer. Cone is an aggregation of sporophylls. Cone is a compact structure obtained by the aggregation of many sporophylls. In leaves, particularly some areas, many sporangias are formed. Such sporangia forming structures are called sporophylls. Yes, this is the cone or strobile. Cone is an aggregation of many sporophylls. Each and every the spherical structures or rounded structures are sporophylls. Such are aggregate to form a conical shaped structure. It is a cone or strobile. In this plant also, these are unmodified leaves, but at the terminal area, some of the leaves are modified into sporophyll, aggregate to form cone. Here also cone. Here normal sporophylls are there, but modified sporophylls are aggregate to form cone. Yes, observe here. This green color entire leaflet structure is sporophyll. How can you differentiate the sporophyll from normal leaf? A normal leaf. In normal leaf, purely green color and helpful for photosynthesis. But sporophyll, green color as well as sporangias also there here. It possesses sporangias. All these brown color patches are sporangias. Due to presence of sporangias, these leaves are called sporophylls. Sporangia producing. Sporophylls possess many sporangia. The purpose of sporangia is production of spores. Okay, spore producing sac is the sporangia. The sporangia producing leaves are called sporophylls. Sporophylls are not only useful for production of spores, also for photosynthesis, because these sporophylls are also like in green color when compared to the normal leaves. These are the sporophylous nature. First time in plant kingdom, sporophyll-like structures are formed in pteridophyte only. First sporophylous plants in plant kingdom, pteridophytes. Try to give answer for these questions. Read each and every option. Pteridophytes are non-embryophytes, embryophytes, archegoniates, trachyophytes, and archegoniates. Choose the correct option. Pteridophytes, embryo formation is there. That's why embryophytes, not non-embryophytes. That's why first one is eliminated. Embryophytes, true. Female sex organs, archegonia, true. Vascular tissues are present, true. Then 2, 3, 4 is right answer. Where it is there? Second option. Yes, good. Second option is the true. Except first remaining all are correct. Actually, pteridophyta, bryo, pterido, gymno, all are embryophytes. Except algae, remaining all are embryophytes. One more question. Trachyophytes among the following are. To answer these questions, but you should learn about a plant group of this example. Lycopodium belong to pteridophytes. Member of the pterido. Pteris is also member of pteridophyte. Polysiphonia, member of algae. Anthoceras, member of bryophytes. You can choose the wise answer based on this given information or data. Trachyophytes among the following. Identify the trachyophytes from following. Lycopodium is a pteridophyta. Pteris is also pteridophyta. Polysiphonia algae. Anthoceras is a member of bryophytes. What is the correct answer? Both bryophytes and algae are atrachyophytes. Pteridophytes are trachyophytes. 1 and 2 correct. Yes, good. First option right answer. First option right answer. Tracheophytic plants, pteridophytes, gymno, angio. But in that 1 and 2 are given as an example for pteridophytes. Next, seed-bearing tracheophytes. Seed-producing tracheophytes. The option should satisfy two characters. One is the seed production, another one vascular tissue presence. Choose. The plant group possesses both seeds as well as vascular tissues. In pteridophytes, no seeds, but vascular tissues are present. In gymno, both are there. Angioles, both seeds and vascular tissues are there. Then right answer is both 2 and 3. How can you eliminate out the pteridophytes? Pteridophyte is actually a trachyophyta, but there is no seeds. Then right answer second option. Okay, good. Correct. Next, fourth question. Pteridophyte includes or identify the members of pteridophytes from following. Identify the members of pteridophytes from following. Answer. Third option. Yes, club mosses, members of pteridophytes. Horsetails and ferns. What about the mosses? You learned in last class. Mosses are example for bryophytes. So except 2 remaining all are correct. Yes. Then the proper prominent answer is third option. Good. Next, one more question. What is the main plant body in pteridophytes? Main plant body in pteridophytes. In algae, main plant body is the gametophyte. Bryophyta, gametophyte. From pteridophyte onwards, main plant body is the sporophyte. Sporophyte is right answer. What are the next question? First, terrestrial plants possess vascular tissues. These are plants also called as the first vascular plants in overall plant kingdom. Answer. Good, good. Pteridophytes are First two land plants are mosses. With vascular tissues, then here we go to pteridophytes only. Good. Second option, right answer. Okay, right. I will explain last group, both gymno and angio in next class. Okay, now bye.
Good morning, students. How are you? So last class we discussed about importance of health, pathogens and immune system, right? So today we will be revisiting one more time. We'll be discussing about fundamental aspects of very important process, very important thing we do. This was the minute classifying difference between a plant and an animal. So Plantae and Animalia were mainly classified based on their ability to locomote, movement. Yes, plants do show movements, but plants do not change their location. They cannot move from one place to other place. But being animals, we exhibit movements as well as we have also this capacity to change our position, to change our location. So this was the one difference between Plantae and Animalia, the other being cell wall in Plantae, no cell wall in Animalia. Plantae performs photosynthesis, Animalia does not perform photosynthesis. Plantae are autotrophic, Animalia are heterotrophic. Many things were there. So at level of DNA, RNA, proteins, cell envelopes, tissue organization, organs, organ systems, anatomy and morphology. At all these locations, we can very clearly discriminate, demarcate between a plant and animal. And one such thing which is helping us to differentiate between plant and animal was again ability to locomote. So you guys need to tell me now, what is locomotion? And compare it with movement. Define locomotion, define movement. First, simple things, movement. So in physics, you might have learnt about scalar and vector. Anything with magnitude is scalar, anything with magnitude and direction is vector, right? Similarly, I can compare movement like a scalar, locomotion like a vector. How? Movement. Any change in your body posture, if there is any difference, any change in your body posture is considered as movement. But if this change in posture also leads to change in your position, location also, that will be referred as locomotion. Any change in body posture could be considered as movement, but if this movement is responsible for change in your location, change in your position, we refer that event as locomotion. Therefore, all locomotions are movements, but all movements are not locomotion. Every locomotion is because of movement, but every movement does not give you locomotion. See, if you wave this hand like this is movement. That doesn't change my location. If I do with same thing with hind limbs, legs, that will change movement. That will be resulting in movement of my limbs as well as locomotion also, right? So all locomotions are because of movements, but every movement does not help in locomotion. Simple thing. And in living world, Monera, Protista, Fungi, Plantae, Animalia. We know this. 5 kingdom. Monera, Protista, Fungi, Plantae, Animalia. Okay. Guys, each and every kingdom from Monera to Animalia, there are members which exhibit locomotion, which do not exhibit locomotion. Yes. Monerans, bacteria possess flagella for locomotion, but not every bacteria was equipped with flagella. No. There might be bacteria without any flagellum. There might be bacteria with many flagella also. Possible. In Protista, chrysophytes, no movement, passive. Dinoflagellates have 2 flagellae. Euglena also have got 2 flagellum. Slime molds, no flagellum. And protozoans, some of them have cilia, some of them have got flagellum, and some of them have got pseudopodia. Therefore, some members of Protista exhibit locomotion, but some members may not be able to exhibit locomotion. Yes, yes. In fungi, as such, an adult growing fungi may not exhibit locomotion, but their spores and gametes may have locomotion, because some fungi have spores and gametes equipped with flagellum. There is chance. In Plantae, adult members, adult algae, well grown, fully grown algae, fully grown bryophytes, pteridophytes, gymnosperms, angiosperms, none of them have locomotion, but in case of algae, spores and gametes have locomotion because they may have flagellum. In bryophytes also, their gametes can move. In pteridophytes also, gametes can move. So there is very limited amount of locomotion in Plantae. Very limited. In Animalia exclusively, each and every member, from sponges, from porifera to chordata. In poriferans, even though the whole animal was attached to a substrate, but their sperms can move. Motile. Even in jellyfish itself is a motile organism. Cnidarias. So all these animal species consist of 1 or 2 cells which can exhibit locomotion. Therefore, locomotion has become a well-defining feature, characteristic of kingdom Animalia. So just narrowing it to our scale, I mean, from Animalia get down to phylum Chordata. In phylum Chordata also get down to class Mammalia. Go to class Mammalia. In mammals also, please focus only on order Primata, that is monkey. Prime apes. We belong to order Primata, we are monkey order. So gibbons, rhesus monkeys, chimpanzees, gorillas, orangutans, all those extinct ancestors, all of us belong to same order, that was Primata. Genus, Homo, species, Sapiens. So just if you narrow your thoughts, only speaking about humans. So inside a human body, yes, humans do exhibit locomotion. Inside human body, there are cells which possess cilia. There are cells which possess flagellum, and human body also possesses cells which possess pseudopodia. Ciliated cells are found in fallopian tubules, trachea, ear, etc. Here inside body, cilia will move, but movement of cilia doesn't give you locomotion. So they will just perform movement. In human body, there was flagellum. Which cell? One cell, males only, spermatozoa or sperms. Sperms may move. So if sperm is moving inside testis of a male person, if sperm moves from testis to urethra also, that movement of sperm doesn't give locomotion to body. So flagellar movement is also just movement in for us. Flagella may exhibit in movement and locomotion in bacteria. Flagella may exhibit movement and locomotion in dinoflagellates. But for us, flagella will be just for movement, not for locomotion. And same time, pseudopodia, macrophages, also known as monocytes, neutrophils. There are some cells in your immune system which possess pseudopodia and which move from one place to other, but movement of these cells doesn't give you locomotion. No, these are for movement only. Then what? Yes, humans possess special set of organs and tissues which help in movement as well as locomotion also. The main tissue which was responsible for movement and locomotion, the main tissue which is responsible for originating movement and locomotion was derived from mesoderm, that was muscle. Muscle gives us ability to contract, relax. Muscle allows us to move. Muscles allow us to convert these movements into locomotion. And muscle was derived from mesoderm. And these muscles might be voluntary controlled by you, or they might be automatically controlled by your brain. As we discussed, sympathetic nervous system, parasympathetic nervous system controls involuntary organs. Somatic nervous system controls voluntary organs. There are muscles which could be controlled by you just by thinking of them, will, voluntarily. But there are muscles, even if you think, you cannot contract, relax them. That should be done by your autonomous nervous system. Muscles are there. So there are voluntary muscles as well as involuntary muscles. The thing was, does muscle perform locomotion all by itself alone? No. If muscle has to listen to you, if muscle has to act according to your will, that muscle must be definitely attached to a bone or cartilage. If a muscle is not attached to bone or cartilage, it may not listen to you. So such muscles will be automatically controlled by your brain. So the specialized tissue or mesoderm derived tissue which is responsible for movement and locomotion of your body was muscle tissue. Done. Now, thing was, muscle. Does every body part consist of same type of muscle? No. Does every muscle look exactly same? No. Does every muscle listen to brain exactly same? No. Does every muscle has same number of nuclei? No. Does every muscle has same color? No. Same number of organelles? No. All knows. But what then? So there was diversity among muscles also. There are some muscles which were attached to bone. There were some muscles which were not attached to bone. So if you just examine muscles under microscope and observe their way of function, if you observe their physiology, depending on their physical appearance as well as physiological functions also, scientists have divided muscle tissues into 3 categories. What were those? The category 1, which will listen to you, these are voluntary skeletal muscles. We call them as voluntary. As they're attached to bone, they are skeletal and they have stripes on them, therefore striped or striated muscles. How do they look? These cells look like long elongated cylinders, and they may have many nuclei, multinucleate condition. What is the synonym for multinucleate condition? Syncytium. And sometimes you can also refer that as cenocytic. Second category. These are not attached to bones, therefore you cannot control them according to your own will. These will be automatically controlled by your brain. Those are involuntary and they do not have any striations, therefore these are non-striated, unstriated muscles. And as they have no striations, no marks on them clearly, they look almost smooth, therefore they are also referred as smooth muscles. They are spindle shaped or fusiform shaped with single nucleus, mononucleate, uninucleate. And third category, these are involuntary, but they possess striations, and this muscle shall never stop contracting and relaxing. The only muscle which was always active in your body, that was cardiac muscle. So how do these muscle cells look under microscope? They look like short cylinders, but they have branches. Short cylinders, but they are branched. So they may have minimum of 1 nucleus to maximum of 2 nucleus. So cardiac muscles are either uninucleated or binucleated. Now, muscle contraction and muscle relaxation is mainly due to proteins. These proteins which help in relaxation and contraction of muscle were actually referred as contractile proteins. So there are 2 types of contractile proteins: thin protein, thick protein. Thin protein is called as actin, actin, actin. Thick protein, myosin, myosin, myosin. So thin actin, thick myosin. Due to regular arrangement of actin and myosin inside a muscle cell, you will be able to see many striations inside muscle cell. Under microscope, you can see these bands. As this muscle has striations, stripes, zebra crossing like marks, we refer that as striated muscle. Same type of striations could also be seen in cardiac muscle. Cardiac muscle is also striated, but not attached to any bone. Therefore, it doesn't listen to your brain according to your own will. It will be automatically controlled by your brain. But no such striations are found in smooth muscle. Smooth muscle does have actin myosin. Guys, smooth muscle also have actin myosin, but arrangement of actin and myosin is not like this. It is randomly arranged, therefore there is no morphological striated appearance in smooth muscle, therefore it looks smooth. And as it was found in inner parts of your body, some doctors also call them as visceral muscles. Done. So voluntary striated, unbranched, no branches. Smooth muscle, no branches, unbranched. Whereas the only muscle to have branching was cardiac, it was branched. See, so voluntary striated, involuntary non-striated, and involuntary cardiac. Three main muscle categories dealt now. Done with this? Guys, I will ask you a question. Try answering this. See, among these three muscles, which muscle has got striations, but this is not voluntary and it is not attached to any bone, and it starts working when our life was just 1 month. We are not even born. This muscle starts working in our body when our age was just 1 month, 28 to 31 days only. What was that? Involuntary, continuous working muscle, cardiac. Very good. Now, tongue. We can twist, turn tongue in many different ways. Tongue was attached to a bone, U-shaped bone referred as hyoid bone. Tongue, if you cut tongue and observe under microscope, will this have branches or not? Tongue muscle, do the process any branching, or is there is no branching in tongue muscle? Is there branched muscle in tongue? Yes or no? Good, no branching, because tongue is voluntary muscle. Voluntary muscles are long cylinders without any branches. Now, yeah, we know cardiac muscle is in heart. Voluntary muscles are biceps, triceps, tongue. Many things are there for voluntary. Now, try giving me 2 examples. Listen to this carefully, guys. Try giving me 2 examples of smooth muscles. Try this. Give me examples of smooth muscles. Where do you feel it? Where do you witness them? Where do you think? Yes, entire stomach, intestine, starting from esophagus till rectum. Smooth muscle is the one responsible for pushing your food through peristalsis movement. Yes, correct. Very good. One more. Entire gastrointestinal tract, common language, alimentary canal, consists of smooth muscle to push food from esophagus to rectum. Yes, correct. Blood vessels, very good. It might be artery or vein. Blood vessels always consist of smooth muscle which will constrict, dilate. Constrict, dilate. Therefore, they can maintain, control blood pressure. Possibly, yes, correct. One more, can you try again? One more. Try this. One more. There is a muscle in your eye which controls how much light enters your eyes. If you're in dark, that part will dilate. If you're in light, this part will constrict. Constriction, dilation. Pupil, yes. Pupil was constricted and dilated because of special muscles called as ciliary muscles attached to lens. Eyes, yes. Ciliary muscles constrict and dilate. They dilate. Dilation means contraction to see more light, which happens in darkness. That's why when you're in dark, your eyes will dilate such that if there is any thin spark of light, you can see this. But if you switch from dark to light very frequently, eyes will pain. You may have experienced this while watching a movie. All of a sudden, you get into a movie theater, your eyes will hurt a bit because outside was light, inside was dark. Therefore, your pupil has to adjust to that low light conditions. Done. Correct. Uterus lining is accepted because uterus lining has myometrium, which helps in birth of a baby. Even labor pains. Labor pain is because of smooth muscle. Now, among these three types of muscles, cardiac may exhibit movement but not locomotion. Smooth muscles help in movement but not locomotion. The only muscle which helps you to move as well as locomote also, the one and only voluntary skeletal striated muscle. Okay, the only muscle to exhibit locomotion for a body was voluntary skeletal striated muscle. Done. And so till this point, so we need to understand, even in case of skeletal muscles also, this was a depth, this is going a bit deep for you guys. Muscles are three types, yes, skeletal, smooth, cardiac. But even in case of skeletal muscle, every skeletal muscle may not look exactly same in terms of color and organelles. Skeletal muscles are also of two types: red fibers, white fibers. What is mean by that? Red fibers, they got more mitochondria in them but less concentration of sarcoplasmic, nothing but smooth endoplasmic reticulum, less reticulum. Whereas white skeletal fibers, they appear white because they got less mitochondria but they got more concentration of smooth endoplasmic reticulum. Red fibers has more mitochondria, white fibers have got less mitochondria. This mitochondria and SCR explains their nature, but color, just like how RBCs consist of hemoglobin, muscle consists of a pigment known as myoglobin. Just like how RBC oxygen transporting function is given to hemoglobin, here oxygen storing function was given to myoglobin. If you have got more myoglobin, more oxygen, more mitochondria, more contraction, no pain easily. Less myoglobin, less mitochondria, less oxygen, you may contract rapidly but it will give you pain. Example, white fibers are found in your eyelids. If you blink eyelids rapidly for 30 times, they will pain. Try that. Blink eyelids, blink continuously for 30-40 times, they will pain immediately. Is it because eyelids usually consist of white fibers? They are fast, but they get fatigue. They are fast, but they get fatigue. Why they are fast? Because they have more SCR. SCR has more calcium. These are fast, but they get exhausted, tired very soon. These are fast, but they get tired very soon. Which English term will you use to replace tiredness? Tiredness, fatigue. If you speak about red fibers, tongue has red fiber. You keep on talking entire day, it will not pain that much, isn't it? Tongue is red fiber. Tongue has red fibers and it has more myoglobin. They are slow. You blink very fast than tongue. Observe that. You cannot move tongue as fast as you are moving your eyelids. Observe this. Tongue is slow. These muscles move slowly, but they do not get tired easily. No, they do not get tired. They do not get fatigue easily. That was their importance. Even biceps, red fibers. Like this. Most of your body parts consist of red fibers. And you can ask me, why meat was red in color? Why chicken looks pinkish red in color, raw chicken? Why raw mutton looks bright red in color? Why fish has white and red pink mixtures? It depends on types of muscles they have. If they have more white fibers, more red fibers, the color of meat definitely changes. That's it. Okay, that's about muscles. And these muscles, guys, always do remember, skeletal muscle cannot function independently without help of a bone or a cartilage. Whenever skeletal muscles contract and relax, this energy must be transferred to bone and bone will perform some mechanical action. This mechanical action leads to movement and possibly locomotion. My question was that, is there any tissue which connects muscle to bone? Is there any connective tissue which is passing energy from muscle to bone? Yes. What was that? Energy from muscle to bone was transferred with the help of dense, regular, connective tissue. What was that called as? Muscle was attached to bone with the help of tendon. How about passing energy from one bone to other bone? Yes, in joints. So energy from this bone must be passed on to next bone through joints. But inside joint also, there was a connection for these two bones. What was that? Ligament. Without tendons, movement is impossible. Without ligaments, locomotion is impossible. If you have tendons, you can pass energy from muscle to bone. If you got ligaments, you can pass energy from bone to bone. So tendon is between muscle bone, ligament is between bone and bone. Okay. And muscles must always pass on their energy to bones. Adult body consists of 206 bones. Some of these bones are found along body axis, and some of these bones are found in limbs. So the entire system of bones of your body was referred as skeletal system. So for our convenience, we can split skeleton into two parts. 80 bones found along your axis. Bones of head, skull, face, ossicles, vertebrae, ribs, sternum. All of these 80 bones are commonly referred as axial skeleton. Bones in the limbs 30, 30, 30, 30. Plus 120 bones plus there are special bones which connect limbs to your body. Those were referred as girdles. 6 bones. 126 bones were there which are referred as appendicular skeleton. So axial 80, appendicular 126. Some of these bones may help in locomotion also, but every bone may not help in locomotion. Ossicles may move but not locomotion. Vertebrae moves and locomotion also. Some of them may exhibit movement and locomotion. Every bone doesn't guarantee you locomotion. Bones of your skull, they're tightly attached, no movement at all, immovable joints. So movement of bone depends on their tightness. So joint is very much important for movement and locomotion. So the most important joint responsible for movement was synovial joint, like this. Between two bones, there must be a gap. See this clearly, I gave a gap. So such joints are responsible for locomotion. We have ball and sockets, hinge joints, gliding joints, saddle joints, pivot joints, many types, 5 to 6 types of joints are there. All of these are helping you to perform locomotion. We'll deal about them in detail once you come here, okay? So at this point, we'll just sign off, and please, all of you, please take good care of yourself. So please stay home, stay hydrated. Temperatures may hit 45, 46 nowadays. So please be very careful, okay? Thank you. We'll meet tomorrow.